أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم لمجموعون إلى ميقات يوم معلوم ثم إنكم أيها الضالون المكذبون لآكلون من شجر من زقوم فمالئون منها البطون فشربون عليه من الحميم فشربون الشرب الهيم صدق الله العلي العظيم. Today I come to tell you about the cilia movement, flagella movement, lecture number 12 for first year class. Interested discussion today once again, reason cilium, flagellum, how it moves, look like it's fiber. Single fiber is there. Look here, if this my finger is cilium, it is moving like this. It is contraction, relaxation, contraction, relaxation. Very simple, that finger is contracting and finger is relaxing. But when you will see biochemistry of this cilium, that is so complicated that I am not telling you all phenomena according to your syllabus. But if you will understand this information which I will give you, you will just think one time that how minor things are so complicated and they are arranged in so sequential manner. So cilium and the flagella is my topic. I am not telling you exactly the complete structure of cilium and flagellum. I am telling how it is moving, with the composition of the cilium which help the cilium and flagella for the movement. Why I am naming cilium and flagella together? Reason, because cilia, flagella, they have the same structure. They have the cilia and flagella same structure in reference to human being, because you wanted to know, sir, how the cilium is related with the human body. Yes, of course, in your cell, in your nose, in your trachea, cilium is present, but not for the locomotion, for some other purpose. And some other purposes you already know, we discussed in respiratory system. But it is a locomotory structure of the flagella, cilia. Amazing. This is the locomotory structure of the paramecium. And flagella is a locomotory structure of the euglena. So these are fiber, just like single structure, single hair-like structure. How they move we are going to discuss today. And you will think that minor thing, how much complicated structure they have. We'll go back to the little information about the cilium and flagellum. We start from the simple information and then we will go to the complex information. Cilium, I will start from the basal body. Cilium and the flagellum, they arise from outgrowth cytoplasm. I am talking about cell. I am talking about the cell. In the cell, there is outgrowth. From the outgrowth it arises, and that outgrowth is called the basal body. And basal body is the area from where cilium and flagellum. Why I am telling cilium? I am not telling cilia. Cilium is a singular and flagellum is a singular. So I have to discuss the single fiber structure. Cilium, flagellum, locomotory structures. Cilium is not the locomotory structure in the human body. What the cilium is doing in your body? What the cilium is doing in your body? In cilium in your body, cilium is involved in trapping the dust particle in the nose. You know, hairs-like structure. Cilium in the trachea is moving the dust particles up, because dust particle will dissolve in the liquid in your trachea. You know the name of the liquid. Of course, the glands which were secreting the liquid was the mucus, and the mucus was secreted by the mucus gland. Mucus gland was secreting the mucus, and then what was happening? It was trapped by dust particles, were trapped by the mucus in the trachea, and the dust particles were moving up by upward movement of cilia. So movement of the cilia is so much important. The movement of the flagellum is so much important. Our body cannot remove the dust particle from the trachea, and our dust particle will move into your alveoli cells, in bronchus, bronchiole. The oxygen way will stop, and respiration may affect, and it never happened due to presence of cilia. But it is present in bacteria. Bacteria, you know, there are some useful bacteria, there are harmful bacteria. Chapters we have to study. We'll go back to basal body. Basal body, from where the cilium arises. Cilium arises. The structure from where the cilium arises. Now I was telling you, this is a so sequential structure, so mannered structure. It is not a simple structure. Look here, 9 fibers, non-microfibrils, 9 microfibrils present. 5 plus 4 equal to 9. 9. And 9 are peripheral 9 are peripheral and 2 present in the center, and 2 centers, 9 plus 2 equal to total how many fibers present. I am not talking about cilia, this is important. I am talking about the one cilium, one small fiber which is a microscopic, not visible. So how a microscopic fiber consists of so technical things in our body, in the microorganisms, and look in the living things. And the cilium consists of total number of the fibers. I have told you how many total number of the fibers are present. 11 fibers are present. Now I will name the fibers now. The name of the fibers is a microfibrils. Total number of the fibers present in the cilium is 11 microfibrils. How they are arranged? Very important, yaar. If you are not attentive, I may stop here. I give you some relaxation, then talk. If you are attentive, I need to talk because this is very important. 11, total 11. Where is the 9? 9 are peripheral, like spokes of the wheel. I will show you figure after some time. 9 peripheral, 2 are the central. 9 plus 2, 11. 9 plus 2, 11. What is the name of these fibers? What is the name? Microfibrils. I am highlighting also. How many microfibrils are present in the one cilium? 11. How many microfibrils are present in the flagellum? 11. How many microfibrils are arranged in a peripheral way? Peripheral means at the sides. How many microfibrils are present in the center? 2. Which microfibrils are more important? The microfibrils which are present in the peripheral side, they are peripheral side, at the side. They are more important because they will contract the cilia. I am talking about one cilia. I am not talking about all cilia of paramecium. Many paramecium, many cilia in the paramecium collectively move the paramecium, but I am talking about the one cilium. How one cilium contract, other cilia contract by same way. So microfibrils, total number of the microfibrils 11. Total number of the microfibrils present in the peripheral site 9. Now one more important point which I just wanted to tell you once again. This is the peak point of today. This is the peak point of today's lecture number 12 for the first year class. I will tell you the microfibrils which are peripheral, which are important, they are not single. Amazing. They are not single. Still they are dividing more. They are doublet. They are the doublet. Look at the doublet. They are doublet. So they are the doublet. So they are in the form of doublet. What is the meaning of the doublet? Meaning of the doublet is that they are arranged in such a form. That they consist of 2 circles. And one circle is outer circle, one circle is inner circle. One circle is outer circle and one circle is inner circle. So I have to tell you about these circles. I am talking about the 2 circles, mean that subfibers, one is a subfiber, one fiber is consist of further 2 subfibers. The 9 peripheral microfibrils, they further consist of 2 fibers, and these 2 fibers are called subfibers. So subfibers, you are watching the subfibers, and now you have to see which fiber is complete, which fiber is not complete. Which fiber is complete? 2 subfibers. Each microfibril consists of 2 subfibers. One is complete, one is not complete. This is the point. One is complete, one is not complete. What I am talking about? I am talking about 11 subfibers. And they are doublet. Why they are doublet? They are doublet in the sense because they consist of the 2 subfibers. When they consist of 2 subfibers, one is a outer subfiber, one is a inner subfiber. Inner subfiber of the doublet. Why it is called the doublet? Because they consist of the 2 subfibers. What is the name of these fibers? Microfibrils. How many microfibrils are there in one cilium? There are 11. How many are present at the peripheral side? 9. How many present in the central side? The central side are present too, but you may be asking, sir, what is the function of the two central fibers? I will tell you later on, but in short I will tell you, they protect the cilium. That is not involved in the locomotion, that is not involved in the movement of the cilia. They are involved in the formation of, they are involved in the strength. They give the strength to the cilium and the flagella. One protein I will name which is not written in the book, and that protein is Naxin, N-A-X-I-N, Naxin. Naxin is the protein which Naxin is the protein which is present in the Naxin is the protein which is present in the cilium to protect. Now you are watching a slide, axoneme. Axoneme, what is the axoneme? One cilium is covered in a membrane. One cilium is covered in a membrane. You can say one complete structure of cilium is axoneme. Axoneme means one cilium is packed in a membrane type structure, and that cilium is called axoneme. So cilium is packed in a structure called axoneme. This is a packet, a fiber, a packet. When you will open the packet, what will happen? When you will open the packet, what will happen? Which structure you will see? You will see the 9 double fibrils. You will see the 9 double fibrils. And 9 double fibrils, 9 doublet fibrils, doublet word written in the book, and the 2 central, 2 single. Look here, the slide. 9 doublet, 9 doublet, you can say the doublet, fibrils present at the peripheral sides, and the 2 single, and the 2 single fibers present at the center. 2 single fibers present in the center. We'll go back to once again the same information which I was giving you. And look at the central fibers, doublet. This was the point I was talking. Now inner subfiber is complete. This is important. I am talking about now one fiber, peripheral, total 11. One cilium, 9 peripheral. Now one out of 9 peripheral I am talking about. It consists of doublet. It consists of 2 subfibers. It consists of the 2 subfibers, outer subfiber and inner subfiber. And inner subfiber is complete. Inner subfiber is a complete circle. I will show you the diagram. Inner subfiber is a complete, and outer subfiber is not complete. Outer subfiber is not complete. It is a C-shaped. C-shaped word you can identify from the cartilage. You know what was happening in the cartilage. The cartilage was having a cartilage is a part of skeletal system present in your trachea, and the trachea C-shaped, that was not complete. That is the part of skeletal system to open the trachea for a long time. So what was happening here? Outer doublet, incomplete. Inner subfiber is the complete. Inner subfiber, and outer is C-shaped. C-shaped means it is not complete. It is not complete. Now, if you will see that inner and each inner subfiber, I am talking about, I think, the base of the topic today. You know that when you watch the movie, you say, what is the name of the hero? If you write any topic, you will write the heading of the topic. If writer will write the drama, he will make the name of the drama. So I am going to name this topic, the structure which is the hero of the cilia, which is involved in the movement of the protein. And I will name this now. You have to be very attentive. Dynein arms. Dynein arms. What is the dynein arms? Dynein is a protein. D-Y-N-E-I-N. Dynein protein. Dynein protein. Dynein arms I was naming, and then I named dynein protein. Why? Dynein arms in the inner subfiber. Dynein arms in the inner subfiber. Why in the inner subfiber? Because the inner subfiber attached with the dynein protein. Dynein arms are the area in the inner subfiber which is attached with the dynein protein. Spelling of the dynein protein, D-Y-N-E-I-N, dynein protein. Dynein protein, important. The name of this drama, the name of this movie, the name of this topic, the headline of this topic, the dynein protein. The structure which is moving the cilium, the dynein protein, D-Y-N-E-I, dynein protein. The hero of this topic, the dynein protein, that is moving the cilium, that is moving the flagellum, and that is attached with the inner subfiber. I will say it is not completely mentioned in this textbook. Technically, dynein protein is attached with both subfibers, the outer subfibers and inner subfibers, and outer subfibers and inner subfibers. I will tell you they slide over each other, by which the cilium contract. I will show you figure, and then you will be able to understand how the dynein protein is. But you will write the dynein protein mostly attached with the inner subfiber, which is complete. Yes, and the area at which the dynein protein will attach, that is the dynein arms. That is called the dynein arms. Now go back to the structure. Now I need your attention, because that will be a key point of today, because you will be able to understand by the figure, and the figure will tell you that how the dynein protein is involving and is making the structure motile. Amazing and interested too. Now look here, these are many cilia. Many cilia are there. I have to highlight. Yes, these are the many cilia. Many cilia are there. Many cilia, one cilia. And you can see the many cilia in the paramecium. This is the one cilia, this is another cilia, this is another cilia, but I am taking the one cilium now. This is the one cilium. So you are taking one cilium. Is that clear? You are taking one cilium. Now one cilium is called as axoneme. Yes, this is axoneme. That was the point I was talking about. Now you will see the structure inside axoneme. It is a circular structure. How many fibers are there? 9. 9 are peripheral. 1, 2, 3, 4. You can count this one. 9, 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 fibrils are there. How many? 2. How many center? 2 in the center. 2 in the center. 2 in the center. So this is the structure, and how they are attached. How they are attached with each other. How they are attached with each other. Spokes. These are the spokes, just like the spokes of the wheel of your motorbike. This is a circle, and these are the spokes. These are the spokes by which they are attached with the central protein, and spokes and the central fiber will give the protection to the cilium and the flagellum. And you already know that the central fibril will give the strength to the cilium, will give the strength to the flagellum, and the peripheral fibrils, which are in the pair form, they are in outer pair form, they are doublet outer and inner form, and they will give the protection, they will give strength to the protein. They will give the strength to the protein. And I will go to another thing which I just wanted to tell you now. If you will be thinking, sir, what is this one? Sir, what is this one? Because you was talking about, they are doublet, but here they are 3. Look here, count here. 1, 2, 3. 1, 2, 3. These are 3. These are 3. These are 3. They are 3. But this is not the structure of the cilium. So sir, what is the structure of this one? Because you was teaching us the structure of the cilium. You was talking about the dynein arm protein present in the cilium. You was talking about the peripheral fibers. You was talking about the central fibers. Now you are talking about, sir, these are the three circles are there. They are basal body. Recall the structure of the centrioles. If you will call the structure of centriole, you will exactly understand this is a basal body. And basal body structure is here, and it consists of 3 microtubules. You know, I was using name 9 triplet microtubules. You remember I was using the 9 triplet microtubules. Why I was naming the 9 triplet microtubules? The reason is that because it consists of the microtubule, the centriole, one centriole consists of 9 triplet microtubule in the center. It means structure of the microtubule is little bit resemble with the structure of the cilium, but cilium I will not tell you that is a complete structure, because that is a doublet. So this is not the structure of the cilium. So that is structure of the centriole. Now you will understand how the cilia is moving. I think by this figure you will be able to understand how the cilium will move. How the cilium will move. Dynein arm. So dynein arms are moving the cilium. And dynein arms are main thing, because dynein arms will move the one cilium. Look here, the figure. Dynein arms. Where the dynein arm situated? In the doublet. Where is the doublet? This is a double. One doublet is this one doublet is this one. How two doublet mean? How two doublets are attached? I am not talking about the cilia once again. I'm talking about the one cilium. One cilium, 9 doublets. One doublet attached with other doublet by help of which thing? By help of red colored dynein protein. Oh, now you got the point. One doublet attached with the other doublet with the red color protein. These are the things nature created. We create also things. The nature create also things. Can you make the machine like that? That one cilium move like this one? And then millions cilium move like this one? And one cilium consists of 11 fibers. And 11 fibers consist of 9 peripheral and 2 central. And one fiber, the peripheral, consists of the doublet. And in the doublet consists of these red color unlimited proteins. And these unlimited proteins are sliding. They are creating the movement. And how they are creating? How they are getting the petrol? From where they are getting the energy? From where they are getting the petrol? They are getting the energy from ATP. What is the ATP? ATP will be hydrolyzed. You know, ATP react with the water to form the ATP, and ATP will react with the water. Yes, hydrolysis, and it will break into ATP, and the phosphate bond of the ATP will be broken down, and phosphate bond, the last 2 bonds of ATP molecules, you know that those 2 bonds are very important because they are releasing the energy, and they got the energy by respiration reaction. You have studied that ATP is the energy rich molecule. After getting the respiration from the glucose, that energy was stored in small packets. I am breaking my ATP molecules to talk with you. So ATP will give you energy, and ATP will give the energy, and this ATP will provide energy by reacting with water. And what happened in this figure? It bends, the cilia bends due to dynein arm, because dynein arms was getting the energy. Dynein arms got the energy, and reason the cilium bend. And this was the information I have to provide you in this lecture. Now we will see one slide once again, because we were watching the slides also. Now this slide, inner subfiber is complete and outer subfibril is incomplete. Now we'll go back to dynein arm is the main protein between outer and inner fibers, and they move the cilia and flagella. Yes, dynein arm, of course, that is the main protein. And we'll go back to next one. The last 4 fibers cause recovery stroke that I will teach you in the movement locomotion caused by the cilia. Thank you very much for listening to the lecture, and I will come with 13 lecture very soon. Allah Hafiz.